new exciting technology called molography, with the promise of not only doing what SPR and BLI can do, but also that it can do it on cells. So let's see. And welcome Andreas Frutiger from Biotech.
Thanks a lot, Stefan. It's a pleasure to be here. I will tell you about how we expand biophysics to in situ. That means complex samples, living cells, with a technology called focal molography. And by this, we should expand the capabilities of label-free interactions beyond what SPR and BLI can do. The Biotech AG, we were ETH Zurich Roche spin-off 5 years ago. Milton bought us 2 years ago. It's a 5,000 people company with worldwide sales force in cell and gene therapy, has many instruments regarding that. I'm Andreas Frutiger. I'm heading the R&D at Biotech, and we are in the biomolecular interaction analysis space, and it's fundamental for the drug development process. There are many technology bubbles that give you this data. SPR, the most important one, then BLI, GCI, and many players in the field. They all use the same principle called refractometric sensing, and they're limited to buffer media and stable conditions. So why do we want to move in situ at all? I mean, there was enough motivation, but actually you might have completely different conditions. You might have mask, you might have reduced effective concentration, you might have elimination. So that's why you want to do this drug discovery with isolated protein, not in buffer, because this can lead to costly late-stage failures, but you want to do it as early on as possible in the real biological system, whether it being a cell or in serum plasma, where it works. So this is an overview of the established technologies. They're very sensitive techniques, SPR, GCI, BLI, known as refractometric sensors, and they're all based on evanescent wave, and that's why they're so sensitive. So they only illuminate the first 100 nanometers above the sensor surface, where the binding event is occurring. And this makes them sensitive because they correlate the refractive index change in this volume to a change in molecules on the surface, and by this construct a binding curve. What is the problem with that? So let's put one of these sensors in a biological environment. Even if you have a non-fouling surface, it usually looks like this. So your sensing volume is full of biological molecules that you do not want to detect, and despite the affinity difference, this is always the case. So it's very challenging to detect the specific interaction. And in addition, temperature needs to be stabilized to 1,000th of a degree. Concentration, buffer changes, all lead to spikes, and you need to process your data afterwards. So state-of-the-art techniques are cross-sensitive. They're very sensitive, but they can only operate in controlled conditions. So what's the solution? We open up a new technology bubble, focal molography or diffractometric sensors, and they will allow to measure in biologically relevant media and in living cells and are stable in situ. They do not see non-specific binding at all. They allow for fast assay development because you can screen your buffers as you'd like. Easy thermodynamics, you can just change the temperature. And it's ideal due to the surface chemistry for DNA-RNA interaction studies, PROTAC RNA as before, and del-Selex hit validation. Why can it do that? So there's actually 3 pillars for a robust and sensitive sensor. And the first one is dark field illumination by evanescent waves. That means you should only illuminate where it's needed. And this is what the established technologies are doing. What they're not doing are the 2 other pillars. They do not make the analyte bind in a submicron pattern. So instead of having a reference here and a signal here, we have an interdigitated reference channel, 1,000 lines next to each other, 200 nanometer lines of proteins. And you need to read out this pattern in a smart way, and we do this by diffraction in Fourier space. It's very similar to what crystallography does. So this is a sensor chip. We have 54 individual sensing areas, and if you zoom in, you see it's not a spot, but it's this line pattern. And the lines are not physically there. They're actually composed of the ligand molecules only. And when you illuminate those lines, each of the ligand molecules scatters a little bit of light, diffracts it in a certain direction due to the pattern, and then when additional mass is binding to this ligand, you increase the amount of diffracted light. And if you zoom out, you will see that all these beams converge in one focal spot, focal molography, and by measuring the intensity of this spot, we know exactly how much mass has bound in the pattern, and in the pattern only. If it's random, it doesn't generate the focal spot. If you turn it, it doesn't generate the focal spot. If you have a gradient in temperature, you don't see it. And as you see, we see an entire array at the same time, so you get 64 sensorgrams in one experiment. How does it look like compared to the state-of-the-art? This is the baseline measurement compared against the surface plasma resonance state-of-the-art device. You see, even if it's double referenced, you have a slight drift over time. And the other device, so molography is fully stable over hours without temperature stabilization. You can have DMSO spikes or glycerol spikes that are almost not visible. And you can even go into 50% human serum in this case, and the running buffer was PBST. So the blue lines are buffer, serum, so no protein, a lot of protein, zero, and again. And you see no response in the baseline, and only if you titrate the target, in this case GFP in, you see nice dose response. You of course can do kinetics in buffer and in spiked human serum. For this interaction it looks quite similar, but we had very different behavior for other interactions when we move to a complex media. You can do rapid thermodynamics. You just ramp temperatures up and down here. These are huge temperature changes, no baseline changes at all below 1 response unit, and you can characterize thermodynamics with it. How do we multiplex? So we have DNA-directed immobilization. We offer everything from 1-plex to 64-plex chips, and there's a single-stranded DNA on each of the spot, which can be different. So that's why you can address them by DNA code. We have conjugation kits with which you can attach a DNA tag to any protein larger than 50 kilodalton, and it gets even easier. We have adapter molecules against Fc, biotin, amine groups, his tags, streptactin tags that you can first inject, and then your sensor is primed to then immobilize your molecule of interest. And the best thing, regeneration is super easy. You can just dehybridize the DNA, and you're ready to go again. Thanks to DNA, it's perfect for DEL-Selex hit validation. So in the Selex or DEL workflow, you incubate your DEL compounds to the protein, and after hit identification, usually the off-DNA synthesis, where you need to narrow down the selection of hits. We propose to expand this and do on-DNA characterization first, because this goes straight forward with a splint ligation approach to our surface, and then only later on move to off-DNA characterization. We have an example here against carbonic anhydrase 9 with these 3 compounds. You immobilize them with 1 injection, and then you get single cycle kinetics within 1 hour. You could also do this with many more. We did actually a couple of DEL screen. The largest one was already 24 that we did on 1 chip. Took only a few hours. And then you get these isoaffinity plots where you nicely can discriminate between compounds. So not only kinetic information, you also can do cross-sensitivity studies. Let's say you don't want to inhibit something in the liver with your compound for sure. You can just flow a liver extract over the chip. If you see binding, maybe you should check again. Or a selection of proteins, because any binding that you see is due to the compound and not to the surface. Then the other one is membrane receptor characterization in living cells. So this technology is actually a combination between flow cytometry and ELISA technology. How does it do that? You can grow a cell culture on top of these chips. We have an incubator. A cell is much larger than the lines, but much smaller than a sensor. So you have many cells per sensor. And in this case, we had a G-protein coupled receptor cell line that was expressing a SNAP tag. And the lines were functionalized with a SNAP substrate. So the cells were sedimenting, and they got immobilized only on the lines. So at the end, we had a GPCR line pattern in the membrane of the living cell. And then you can actually do an assay just on this GPCR, and you're insensitive to what happens to the rest of the cell without any labels. So you only see when you stimulate this one and inhibit this GPCR, what is happening. You can compare it to bread, you get KD values from it, and so on and so forth. It cannot only do this, okay, oh, sorry. It cannot only do this. We have the spot intensity, which is this very specific channel, which can also, with a nearby monogram, then detect secreted protein, like cytokines, when you stimulate or activate cells. You can measure, like a refractometric channel, SPR-like, for mass redistribution and so on, when you observe the spot movement, and this comes for free. And we have a fluorescence channel built in. So this is our Max Matchmaker. It's outside in the booth, the only instrument that incorporates focal molography. We'll tell you more about it later. We have already 12 devices in the field at 7 customers. And as a conclusion, it's multiplex, it's automatable, drift-free, sensitive, label-free, and it works in complex samples. Save the date if you want to come to Zurich. And with this, I thank you for your attention. There's a poster from Sylvia Huber, this drug discovery for endogenous proteins from Hoffmann La Roche outside, and Sophia and myself are welcoming you to the booth in case you would like to know more. Thank you.